Hi guys, let's talk about enzymes and how they aid digestion. Now, an enzyme is something that all living things have. Even fruits have enzymes in them. And an enzyme has a very specific job in living things. It aids and speeds up biological reactions. That means it either helps break bonds in molecules or it helps build bonds in between molecules. So we're going to be talking about the enzymes that are specifically in your digestive tract. If you look at this sheet right here, we have set up the digestive tract with 3 separate enzymes. So these down here are enzymes, and there's 3 of them, okay? These right here represent the molecules of food and nutrients that you put in your body when you eat. They're known as substrates, okay? Now, enzymes are designed to work with one specific kind of substrate. So when we are trying to break bonds or build bonds, we have to make sure that we're working with the right things. So if we look at this first enzyme right here, we need to find the substrate that matches with its active site. This part right here, where the substrate fits into an enzyme, is called an active site. Now we could build a substrate that would fit, but this one right here is already ready to go, and it's going to fill that active site. When an enzyme and a substrate come together and bond, it's called the substrate enzyme complex. Okay? So when they're together, it's the complex. Now it's going to do work on that enzyme breaking those bonds. Here's another enzyme, and we're going to try to find the perfect fit. And so we're creating another substrate enzyme complex. Okay? Now, when the enzyme is done with the substrate, it's going to release what it has. And in this case, it was breaking bonds. And so those 2 pieces got broken apart, and we now have our product. 2 separate molecules that came from one molecule. Now, enzymes not only break things apart, but it also puts bonds together. So in this case, this active site is designed to work with a whole bunch of these little green molecules. So we're going to put them in there, and we're going to fill the active site. Okay. Now, the product of this is going to be one molecule that's going to have the same shape like this. So when this enzyme is done with these substrates, it releases them into one product. So in this case, our product is one molecule made from many. So the product could be 2 molecules made from one substrate, or it can be one product made from several substrates. Okay, so it can go both ways. Now, sometimes we get something called an inhibitor. Now, an inhibitor gets in the way of an enzyme doing its job. Okay, forgot to write that one down, so here we are. Now, this guy is an inhibitor. It's going to prevent an enzyme from doing its job. It's going to get in the way. So when I put the inhibitor on the active site, it's called a competitive inhibitor, because now this substrate that wants to use this enzyme can't do it anymore. He can't use it because somebody else is in the way, and that prevents the enzyme from doing its proper job, and it prevents the substrate from doing its biological reaction. Now, eventually, this substrate could split apart into its many pieces on its own, but this enzyme is what speeds it up and makes it useful. So this competitive inhibitor is preventing the substrate from getting to the active site. The other kind of inhibitor is the non-competitive type. So the non-competitive inhibitor doesn't go to the active site, but kind of attaches itself to the enzyme in a way that it can give the enzyme some wrong instructions, or just generally kind of confuse the enzyme into what it's supposed to be doing. So the substrate can fit in there and create the complex, But because it's attached to this inhibitor, it's not going to work as well as it used to. Okay, so let's review. The substrates are all the nutrients and molecules within the nutrients that you eat. Okay, enzymes are what speed up biological reactions. They can either break bonds or build bonds. The complex is when an enzyme bonds with a molecule or a substrate and it starts to do work on it. The product is what you get when the enzyme releases the substrate and the bonds have been broken or the bonds have been built. And our inhibitor gets in the way of work getting done. Okay? If you have any questions, don't forget to ask and don't forget to take your notes.